Okay, I'm going to share my screen. Okay, we'll just go here first. Okay, so guys, can you hear me كويس? Yes, okay, good. Okay, so for this lecture, first of all, you can see that I use the same lecture that you guys have, that the doctor gave you, because I wanted, I don't like using new lectures in entirely different PowerPoint. I want you guys to be able to study from this lecture too, so that's useful for you. And I also, the only changes I did is add my own notes to the lecture to make it easier, add my explanations to the notes right here as well, notes section, and I added some pictures as well, okay? That's the first thing I want to mention. The other thing is that I want to make this lecture interactive, so I'm going to be asking you guys questions, and you don't have to unmute yourselves. You can even type in the chat, okay? Even if you don't know Adi, you can just say you don't know. And yeah, that's it. I think we'll start right now. Okay, so what do you guys know about cholesterol? Can you tell me that? What comes to your mind when you think of cholesterol? Any diseases, any functions of cholesterol that you know of? None of you guys know anything about cholesterol? Aren't you like first years? Okay. Okay, so cholesterol, the first thing that you need to know is that cholesterol genuinely has a bad reputation because it's linked to heart disease, it's linked to atherosclerosis, it's linked to many diseases that we have, okay? And we know that high levels of cholesterol in the blood is actually a risk factor to many diseases, okay? But it has vital functions in our body, and this is important for you to know, okay? And that's mainly what we're going to be discussing, their functions and how the cholesterol was made and synthesized, okay? So, first of all, it's a membrane component. So it's a component of the cell's membranes, and the purpose of it being there is because it allows cells to be, it's not stiff, they can move better, it provides fluidity to the cells. The second of all, it's a precursor to many different molecules like bile acids, steroids, and vitamin D, okay? Have you guys took GI, صح? So can you tell me what bile is? One person, just one person, tell me at least what you know about bile. Emulsifies fat. Yep. I like how you use the exact word command on the slide. It's good. Shows at least you're reading. Yeah, so someone directly messaged emulsifies fat. That's good. So you need it for, because we know fat is lipid soluble. So it's good. In order to be able to digest it and absorb it, you need bile, okay? And from where do you get the bile? You have to synthesize it from cholesterol. Vitamin D, we know that we can synthesize vitamin D in our body. And in order to synthesize vitamin D, we need cholesterol. It's the very first precursor of vitamin D. And vitamin D is involved in calcium metabolism. So anything related to calcium, it helps with calcium reabsorption from the urine and from the intestine. It basically helps you have a good amount of calcium in your body. Okay, let's just leave it at that because it has a lot of functions as well. Steroid hormones, this we're going to go deep in detail and actual diseases about it. You have androgens, you have estrogens, corticosteroids, and glucocorticoids. Androgens and glucocorticoids are technically under corticosteroids. That's just one class. It's one class of steroids that are formed by the adrenal glands, okay? So androgens are under corticosteroids and glucocorticoids are under corticosteroids, okay? Technically, they all fall under the same heading. But yeah, we'll talk about the functions of each towards the end of the lecture. Now, properties of cholesterol. So you know that cholesterol is lipid soluble. What does that mean? Okay, what does it mean for a cholesterol? What does it mean if I say cholesterol is lipid soluble? Can anyone guess? حتى just suggest an answer, even if it's a stupid answer. It can get dissolved in lipids, yes. What about in blood? Can it exist? Can it be present freely in blood? No, yeah, it can't be. Okay, because it's hydrophobic, it has to be anything. Think of it as a rule. Anything that's lipid soluble cannot exist freely in the blood or any fluid until it's bound to something like a protein, like a carrier protein or something, or packaged. In case of cholesterol, it gets packaged into lipoproteins. Did you guys take your lipoprotein lecture? Well, not yet. You took it. So you know like what lipoproteins are. We have VLDL, HDL, and all. So these lipoproteins, they carry cholesterol in order to allow cholesterol to be transported from a tissue to another tissue. Because we know cholesterol is lipid soluble, it can't flow freely, okay? And the ones that are free, they have to be bound to something, to some sort of protein or anything to allow it to be transported in the blood. Okay. Now, another thing you need to know is the process of esterification, okay? So this is the cholesterol molecule. You can see it's kind of like a ring structure, it has multiple rings, and this is the side chain, and it has a hydroxyl group here. So in the process of esterification, it's basically when a fatty acid combines with this cholesterol at this site, okay? And this makes the cholesterol even more lipid soluble. So can you guys guess why you would want to do that? Why would your body want to make cholesterol esters, which are more lipid soluble? Does anyone know? Or can guess? Why would you want cholesterol esters? Okay, I'm guessing you guys don't know. Alas, we'll see. So, to be packaged inside the lipoprotein. Yeah, basically, to be packaged in a more efficient manner. So I added this picture for your convenience to explain better. So this is discoid. This is a discoid HDL. Discoid basically means that this shape is a discoid, okay? So this is HDL that absorbed cholesterol, but the cholesterol it's in normal form, okay? This isn't a cholesterol ester. And as you can see, as you can see, this is the yellow dots, these are the cholesterol. Because they're partly hydrophilic, okay, they're stuck in the outer parts, okay? They're not completely hydrophobic, okay? So that's why they can stick, you know, in the membrane happily. They're not that bothered, okay? Once they become a cholesterol ester, that's when they become so hydrophobic that they become packed inside, okay? Once they're packed inside, this allows this makes the HDL go from discoid to spherical, which allows more cholesterol to be packaged inside. So basically, this spherical HDL is able to transport more cholesterol. And that's why you need cholesterol esters, because they help improve the transport of cholesterol, okay? They become more packaged with more cholesterol, basically, okay? And as I mentioned, where does the link happen? At this place, okay? At the hydroxyl group, at the hydroxyl site. And this is the fatty acid that combines with it, okay? Let me remove this, okay. Now, okay, sources of hepatic cholesterol. I added this picture just to better illustrate this. So where we know that the first thing, the most important thing you need to know about cholesterol is that most of the cholesterol you have in your body and you need in your body is made by the liver, okay? And so most of the cholesterol, as I mentioned, is formed by de novo synthesis. De novo synthesis means you create it from scratch in your body, okay? Other parts, let's say other sources of cholesterol is the dietary cholesterol you eat, okay? And we know that if you remember from GI, the dietary cholesterol you take in, it gets absorbed through the enterocytes. Once they go through the enterocyte, the cells lining your GI tract, they go into the lymph. And as soon as they're absorbed, they're put into chylomicron remnants, because they can't be, because as I said, they're lipid soluble, so they can't be transported freely. So they're put into chylomicron remnants, and the chylomicron remnants flow through your blood and then end up coming to your liver, okay? And sorry, once they're absorbed in your enterocytes, they're not called chylomicron remnants, they're called chylomicrons. And then once they end up reaching the liver, they're called chylomicron remnants, okay? So first it's called chylomicron, then it gets transported through your blood, goes to your liver and becomes, and once it reaches the liver, by then it's called a chylomicron remnant, okay? And so that's one source that comes from your dietary cholesterol. Then you also have cholesterol that's synthesized from other tissues in your body, extrahepatic tissues, okay? So all this cholesterol comes to your liver, stored in your liver, and then goes where? It's either secreted as free cholesterol in the bile, or it's converted to bile acids and salts. So the bile that you have that is secreted into your intestine whenever you eat something and you have bile, if you look at the bile, it has bile salts and bile acids and free cholesterol. And the bile salts and bile acids, they're made from the cholesterol, okay? Can you please explain what exactly is a chylomicron? Okay, the best way to explain this is to add this picture. I didn't add it صراحة من أول because it was شوية. Okay, one sec, let me do this. I'll do it full screen. Okay. Okay, ignore the background, look at this. This is your intestine, okay? This is your intestine. You have here cholesterol that comes in, okay? As soon as the cholesterol is here in your intestine, it gets absorbed into your lymphatics. So this is a lymphatic vessel, and then it goes through your blood, okay? At this point, once it's absorbed, it becomes a chylomicron. It's packaged into a chylomicron. This is a chylomicron. It's like this package, okay? It's a package made of, I think, protein and other substances. And inside of it is the cholesterol. Then it flows through your blood. So it's flowing through your blood and it becomes a chylomicron remnant. When does it become a chylomicron remnant? When a lipoprotein lipase takes some of the triglycerides or fats inside the chylomicron, okay? This is the step I didn't want to explain so that you don't, it's not the focus of the lecture, that's why I didn't really focus on it. But just for your understanding, the chylomicron is formed as soon as you absorb the lipids from your intestine. Then once it interacts with a lipoprotein lipase, which is found on in the walls of your blood vessels and tissues, it becomes a chylomicron remnant, okay? And the remnant just has, the difference between a chylomicron remnant and a chylomicron is that the remnant has less cholesterol, okay, because it's been absorbed. It's been broken down by the lipase. Okay, one second, let me, how do I get this? Okay, you can see the screen, صح? We're back to the same screen. I just want to make sure you don't, okay, good. All right, so that's the chylomicron remnant. We were saying the cholesterol in the liver, once the liver makes the cholesterol and takes it from all the other sources, we said we either convert that cholesterol into bile, okay, or the cholesterol is secreted with the bile as is, or it's packaged into VLDLs, and then it goes through to your blood, okay? So bile goes into your intestine and the VLDL goes through your blood. And these are all just facts that you need to know and memorize. Dietary cholesterol, you need to remember that dietary cholesterol does not affect the level of the cholesterol in your blood as much. It's mainly because the main source of cholesterol in our body comes from the liver. It doesn't come from our diet. So this is one thing you need to keep in mind. That's why someone who has very high triglyceride level of triglycerides, you can't just tell them, okay, just decrease the level of cholesterol you eat and you're good. It doesn't work like that. They have to take a certain drug that inhibits the synthesis of cholesterol in their body in order to have low cholesterol, okay? And okay, so this is, I mentioned it. Okay, now dietary cholesterol. Yeah, this is what I said. And I think you need to know these numbers, that this basically shows how in de novo synthesis, it forms most of your cholesterol. Like 800 milligrams per day you get is from de novo synthesis. And 1000 milligrams is excreted. And they're all excreted how? Through bile. Okay, that's the only way that you can excrete cholesterol. And like I said, lowering cholesterol in your diet has very little effect on the blood cholesterol. Okay, this is pretty much the same. This is liver, VLDL. This is how it gets transported to peripheral tissues. If you have any questions about these, tell me, but I'm trying to stick to what's high yield. Okay, so now cholesterol synthesis, okay? Now we're going to talk about the de novo synthesis, the process of making cholesterol. The first starting point, in order for you to make cholesterol, you need acetyl-CoA, okay? Okay, so someone's asking, do the two enzymes in the previous slide do the same thing? Okay, lecithin, cholesterol acetyltransferase, and cholesterol acetyltransferase protein. I don't think so, because cholesterol, LCAT, it converts the cholesterol in your peripheral tissues. It's converted to cholesterol esters. That's what I'm getting at. And the cholesterol ester transfer protein, I think it's just for converting HDL to VLDL. But you need to make sure, honestly, you should check the lipoprotein lecture, okay? Because I think this has more to do with the lipoprotein lecture, okay? Yeah, she explained it. I don't know who S is, I'm sorry, but she, like she said, the cholesterol is converted to cholesterol esters using the LCAT, okay? And I think the CETP, cholesterol ester transfer protein, it just transports the cholesterol here to the VLDL, if I'm not mistaken. Okay. All right. So, as I was saying, for cholesterol synthesis, your starting point is acetyl-CoA. Okay. Do you guys remember any pathway that forms acetyl-CoA? Any pathway that forms acetyl-CoA? Do you guys remember anything? Yeah, TCA. Good. You can also follow, fatty acid oxidation, fatty beta oxidation. Exactly. So these are some sources. So these processes, you take from these processes acetyl-CoA. Then the acetyl-CoA goes through multiple steps, I will see, to form the cholesterol. What you need to know is, other than acetyl-CoA, you need two more things. Do you guys remember what those things are for de novo synthesis of cholesterol? You need acetyl-CoA and two more important molecules, NADPH and ATP. Yep. Okay. So what you want you to know here as well is that 80% of this process happens in the liver. Okay, so de novo synthesis happens in liver. It can also happen in the intestine and skin. What you need to know is just, it's mainly, where does the de novo synthesis happen? Mainly in the liver cell cytoplasm, okay? And these are the three that I mentioned, the NADPH, ATP, and acetyl-CoA. And like you guys said, it comes from beta oxidation of fatty acids, also dehydrogenation of pyruvate, when pyruvate undergoes dehydrogenation, and oxidation of ketogenic amino acids, okay? Do you guys know any ketogenic amino acids? Do you remember any of them? I don't know if you took it yet, because the mole lectures are, I don't know if you have the same mole lectures the way the organization we had. We took it, but it's not in the midterm. Okay. Yeah, leucine, okay. So yeah, you have leucine, lysine, tryptophan, and tyrosine. Okay, these four, they're all in the notes, by the way, here. I added the explanation for them, okay? Just so you have it, you keep it in mind. So yeah, you can use those to make acetyl-CoA. You have NADPH, it's need for as a reducing power, and we'll see it, it's better when we go through the steps, yeah. Okay, so we said primary site liver, these are the secondary sites. These sites right here, adrenal cortex, ovaries, and testes. They form cholesterol in order to form steroids, okay? That's why they form cholesterol. So there's steroidogenic tissue that form cholesterol in order to form steroids, and we'll talk about the process as well. And this is the overall equation. Do you guys understand why it's like this? 18 acetyl-CoA, 18 ATP, and 16 NADPH. So it's because basically you have the acetyl-CoA, you need 3 acetyl-CoAs to make one HMG-CoA. And then the HMG-CoA is converted to mevalonic acid, and in order to continue, you need 6 mevalonic acid. So in order to make 1 mevalonic acid, you needed 3 acetyl-CoA, okay? So if you want to make 6 mevalonic acid, you'll need 18 acetyl-CoA, okay? And you'll understand this better when you see this, okay? So there's 4 main stages. And what the first stage, as I mentioned, is the phase where you have HMG-CoA formation. My friends and I like to call it HMG-CoA only because it's easier. So if I refer to it as that, just know that I'm referring to HMG-CoA, okay? So the first step is two acetyl-CoAs combine, and then another acetyl-CoA comes and it combines with those, and then it forms a HMG-CoA, all right? Then the HMG-CoA becomes mevalonic acid, which is what I mentioned in the previous slide. And in order to do that, it needs to be catalyzed by an important enzyme. So this enzyme needs to be available, which is the HMG-CoA reductase, Okay, do you guys know anything about this enzyme? Or is this the first time you heard? Like, what's important about this enzyme? It's inhibited by statins, yes. It's rate limiting, right? So it's the rate limiting enzyme in this reaction, in this pathway. And what rate limiting means is basically in every chemical reaction or pathway, there's always a rate limiting step, which is the slowest step in the pathway. And it's dependent on, and in this case, it's this step, with the one that forms mevalonic acid from HMG-CoA. And in order to do that, it needs the HMG-CoA reductase, which is regulated by multiple different factors that we'll talk about. And so if this HMG-CoA is inhibited, then this whole pathway basically cannot happen, okay? All right, so this is one thing. So as I said, the second stage is when the HMG-CoA forms the mevalonic acid, okay? Here he has it, I refer to it as second stage, but here he says the stage 2, it converts C6 to C5 intermediate. So C6 is the mevalonic acid, and it gets converted to C5. So here, the stage he's referring to is this one, okay? And I just said this second stage just so, because it's part of the pathway, okay? But this is what he considered the second stage. Mevalonic acid to isopentenyl pyrophosphate and dimethylyl pyrophosphate, okay? One thing you need to know about this step is that it needs 2 NADPH. That's why, because HMG-CoA, in order to become mevalonic acid, it needs to be reduced. And in order to do that, you need NADPH, okay? So that's why we need NADPH in the beginning. It's for this step. So always remember, for the important steps, always remember the enzyme responsible for it and the molecule required, like if there's an NADPH or ATP that's required, because they ask about this stuff. So then the mevalonic acid is converted to isopentenyl pyrophosphate and the way this happens is that first it undergoes phosphorylation, okay? And it undergoes phosphorylation 2 times. So you have 3 ATP molecules right now, صح? 2 of them are used to phosphorylate mevalonic acid. The last one is used to decarboxylate the intermediate, okay? That's why it went from a C6 to a C5, because it got decarboxylated. That means a carbon atom was removed, okay? So it got phosphorylated twice. So it has, this, if you look at this, the structure of this molecule, it has 2 phosphate groups. And it was decarboxylated, so it's C5. It's 5 carbon atoms, not 6. That's why you needed the 3 ATPs. And you need to know that this isopentenyl pyrophosphate, it's an activated isoprenoid, and it's in equilibrium with dimethylyl pyrophosphate, okay? They're formed in equilibrium, okay? And this image kind of shows it better. You see how this is mevalonic acid. It forms, this is an intermediate, okay, that becomes decarboxylated. You can see here a carbon atom is leaving, okay? And it forms isopentenyl pyrophosphate. And this isopentenyl pyrophosphate is in equilibrium with the dimethylyl pyrophosphate. And this, the irreversible, how it's, the arrows showing that it's a reversible reaction helps kind of remember it, because it's the only reversible reaction in the whole thing so far. So that's why they're in equilibrium, okay? And both of them are activated isoprenoids. Okay. So that was the second stage, forming this isoprenoid. Then the 6 isoprenoids end up forming the squalene, okay, or squalene, okay, which is here. This is the squalene, okay? Squalene is C30. That's why we needed 6 isoprenoids. So how did they form exactly? Let me show you, Hina. Okay, so you see here, this is the dimethyl pyrophosphate, and then it forms the geranyl pyrophosphate. And that's because if you look at, where was it? I want to remember exactly which isoprenes they form. Yeah, the two isoprenes, they're condensed. Which two isoprenes? The ones in equilibrium. So isopentenyl pyrophosphate and dimethyllyl pyrophosphate, they join to form geranyl pyrophosphate, which is C10, because each of them is C5. Then the geranyl pyrophosphate joins with another isopentenyl pyrophosphate, forming farnesyl pyrophosphate, which is C15, because 10 plus one of these, which is 5, it forms a C carbon 15 molecule, which is farnesyl pyrophosphate. Then farnesyl pyrophosphate joins with another farnesyl pyrophosphate, and that makes a 30 carbon molecule, and that's called squalene. Okay. And okay, so now the last step, which is the cyclization of squalene. Okay. It's an interesting thing about this step is that because it's so complex, like many scientists don't even know how it happens exactly, and they studied it for many years because it's so fascinating. And what happens is basically this linear structure, okay, and it ends up being like this squalene, it ends up being this cholesterol shape because it undergoes cyclization, which means it basically forms these rings. And it's actually like, I think there's like 20 catalyzed steps in between to form this cholesterol from the squalene. There's like 20 steps, and you know, obviously, alhamdulillah, we don't need to know it, and you won't need to know it for your exam. But I think the only thing you need to know is that squalene it becomes cyclized or whatever to form the cholesterol. Okay. If there's anything you don't get, let me know. I think I mentioned everything that's important here. So the last stage, this stage is just needed for the shape of the cholesterol to bring the ring structure shape together. And here I highlighted the ATP just to show you how it was used. The first two steps were to cause phosphorylation of the mevalonic acid catalyzed by kinases. Whenever you see kinase, that means it's an enzyme that causes phosphorylation. And then decarboxylated, needed an ATP to decarboxylate it. Okay. Now let's see this. So now regulation of the enzyme. So as I said, there's a key regulatory enzyme, which is hemage-CoA reductase, and it regulates the rate limiting step. And there's 3 ways it can be regulated: by covalent modification, degradation of the enzyme, and gene expression. Do you guys know what covalent modification is? No one knows what covalent modification. Do you know any other enzyme that can be covalently modified? Okay. Okay, yeah, okay. Phosphorylation either activates or deactivates an enzyme. That's an example of covalent modification. So it's basically when you modify a molecule by forming some sort of covalent bond or removing a bond that ends up making the whatever enzyme that's modified either active or either activated or deactivated. Okay. So for HMG-CoA, it's actually it's active when it's dephosphorylated. Okay, and we'll talk about it. Let's just go straight to the slide. Yeah, this one, HMG-CoA reductase. As you can see, this is the inactive form, and this is the active form. In order for the inactive form to be activated, it needs protein phosphatase. And when I said kinases, they cause phosphorylation. Okay, phosphatase, you should memorize it like your name. Phosphatase means dephosphorylation. Even if you don't know the name of the enzyme, but you see phosphatase in the end, that most likely means this enzyme causes dephosphorylation. Okay. So protein phosphatase, it dephosphorylates HMG-CoA to activate it. And protein kinase A causes phosphorylation, so it inhibits, it causes it to be inactive. And what induces protein phosphatase is insulin. Can you guys guess why would insulin stimulate HMG-CoA? If it stimulates HMG-CoA, that means you want cholesterol synthesis. صح? why would you want cholesterol synthesis? Yeah, why would insulin want cholesterol synthesis? Why would it cause it? Can you guys guess? It's related to a state we're in after that has high insulin or something. I don't know. Should I re-ask the question? I feel like I explained it badly. Just can you guys guess why would insulin? Okay.
After a meal.
Yeah, after a meal. Exactly. So after a meal, why would you? Okay, so after a meal, you have high insulin. Why would you want cholesterol to be formed after a meal? Okay, for bile synthesis. Exactly. Because you need bile in order to emulsify the fat that you just ate. Okay, most of the food we eat has cholesterol. Okay, so you need to emulsify the fat that's in the food, and that's why you need form insulin promotes cholesterol synthesis to make the bile. Okay, that's how I remember it. That's how I remember. It helps me remember, insulin. Okay, it's the one that promotes HMG-CoA to work. Okay, and how does HMG-CoA work? By dephosphorylation. Okay, this is 100%. Definitely, they're probably going to ask you about it because they love asking about dephosphorylation and phosphorylation in more. Okay, so yeah, I think I mentioned everything. Feeding promotes cholesterol. Yeah, we said that. Okay, and other things that the feeding provides. Whenever you eat, what happens? It activates the regulatory enzymes, which I just said, and it also provides a lot of acetyl-CoA from the food, and it increases NADPH production. So these you just need to remember. Okay, now for inhibition. So whenever insulin does something, always remember that glucagon does the opposite. Okay, they're always polar opposite. So insulin decreases the blood glucose levels. Glucagon increases blood glucose levels. Insulin activates HMG-CoA. Glucagon deactivates HMG-CoA. Okay, but it does that in a more complex step. It's not direct like this one. So glucagon causes stimulation of cyclic AMP. And if you guys know, you should have the cyclic AMP pathway memorized, okay? That whenever cyclic AMP is activated, the first thing what it activates is protein kinase A, okay? And do you guys know exactly which enzyme causes production of cyclic AMP? This you should know as well, Yani. Does anyone remember what enzyme forms? It's in the membrane, or it's related to a receptor, Yani, usually. Adenylyl cyclase, exactly. Okay. So adenylyl cyclase is, any enzyme or any substance that binds to adenylyl cyclase or causes adenylyl cyclase activation. Directly, adenylyl cyclase increases cyclic AMP. Then the cyclic AMP activates protein kinase A. Okay. Then the protein kinase A phosphorylates it, inhibiting it. Okay. Okay. Do you guys want to have a break after we finish with cholesterol synthesis, or do you want me to continue till the end of the lecture? Whether you don't mind, continue, please. Okay, خلاص. We'll continue to the end. Okay. طيب. What's the next? Okay, so this is another way of regulating cholesterol synthesis. You have transcriptional control. And there's another thing. Where is it? Okay. So as soon as the cholesterol levels, they rise in your cell, in any cell, okay? What happens is that first HMG-CoA reductase is decreased by degradation. So we know HMG-CoA has a half-life. Any molecule or any enzyme usually has a half-life, okay? Protein. So its half-life is 2 to 3 hours. And every time when your cholesterol levels inside the cell increase, its half-life gets shorter in order to degrade it faster, okay? As a way to regulate the cholesterol levels in your cells. Another thing is that this enzyme ACAT, which is the acetyl coenzyme acetyltransferase, it increases in order to promote storage of the cholesterol so that there's decreased levels. And the last thing is that the LDL receptor synthesis, so the gene responsible for making LDL receptor, okay, it decreases. How this would help is that the LDL receptors, they're usually found here on the outer side of the cells, and they promote LDL uptake of fat. So they promote absorption of fat from LDL in your blood to go inside your cell. You don't want that. When the cholesterol levels are high already inside the cell, you don't want that. So you decrease the amount of LDL receptors. Okay, so these are other 3 ways that are related. I mentioned them all كمان هنا in detail. Yeah, it's basically what I mentioned. So cholesterol degradation, it cannot be broken. Yeah, this is how, this slide's more about how you get rid of cholesterol in your body. As I mentioned, the main way of getting rid of cholesterol, or I think it's the only way of getting rid of cholesterol in your body, is through bile, okay? So the cholesterol either gets converted to bile salts or it's released in the free form in bile, and then it goes to your intestine. In your intestine, some of it gets reabsorbed as bile acids, and some of them get excreted with feces. Okay, and that's the only way you can be done basically with cholesterol. So what do you think could help? Should I ask it now or should I ask it? I'll ask it later because I don't know if you'll get it right now. Okay, heart disease. So this is what we link, most people link cholesterol with, okay? We know that many of the diseases of the heart are because of high cholesterol levels. Either atherosclerosis and coronary heart disease, it's all because of cholesterol being piled up. Yeah, and here they're saying, he's saying the cholesterol levels, they're directly related to the heart disease. And as the cholesterol level increases, your risk of getting heart disease, and if you already have a heart disease, it becomes more severe, basically, okay? What I'm guessing he means here, because I was contemplating what he means exactly by this, I'm guessing he means that as you decrease the level of cholesterol in your blood, there's less risk of morbidity and mortality, okay? So yeah, he's just showing basically that cholesterol is good, but at very high levels, it's obviously not good. And I'm sure you guys know that. Okay, yeah, this is the drug we mentioned earlier, because we said, you know, changing your dietary diet isn't the only thing that can help with, it has little effect on your blood cholesterol. You need to inhibit the main source of cholesterol, which is the hepatic synthesis of cholesterol. That's why they found the drug statins, or formed the drug statins, okay? This is a picture for statins. I like this one better because it shows clearly how it looks. So this is Simvastatin. It's one of the statins. And this is the HMG-CoA. You can see that it's kind of similar. Okay, it's a structural analog. And the purpose of Simvastatins being structurally similar is that it can bind to HMG-CoA and basically occupy it so that it doesn't go and perform its purpose, which is converting the we forgot the intermediate. It's the 3-acetyl, the 3-carbon molecule into HMG-CoA, or was it HMG-CoA? Wait 1 second. I got a brain freeze, معلش. Yeah, it's converting HMG-CoA to mevalonic acid. So you want to prevent this step by what? Occupying this enzyme, okay? So occupying it with what? With the drug, the statin, so that it doesn't bind to HMG-CoA and convert it to mevalonic acid. Okay, is that clear? Okay, good. So yeah, it's basically a competitive inhibitor, as in it competes with HMG-CoA. So the enzyme will bind to whatever is available. صح? So if you have a lot of statins available, it's going to bind with the statin and then forget about the HMG-CoA. That's how I remember it. Okay, that's what competitive inhibition means. Okay, they compete with each other. And another function of statins is that it decreases the LDL cholesterol, okay? So it causes 20 to 40% decrease in the cholesterol and LDL, and it increases the LDL receptors. And we mentioned in LDL receptors, or I mentioned that the LDL receptors, they cause uptake of cholesterol from the blood into the cells, okay? So by doing that, you're decreasing the levels of cholesterol in your blood. So this is one way, another way actually, of how statins decrease the level of cholesterol, okay? So 3 ways: inhibiting HMG-CoA, decreasing LDL cholesterol, and increasing the uptake of LDL, okay? I think that's mentioned everything. Okay, so this is a bit of a clinical scenario. Familial hypercholesterolemia. So familial hypercholesterolemia, in its name, it's a genetic condition of really high cholesterol levels. And it's, okay, can you repeat the last part? Okay, so are you talking about the functions of statins? Exactly, yes, okay. Okay, so statins, you know how I mentioned Hina? Where is it? Hina. This is a cell, okay, and you have an LDL receptors on this wall. LDL receptors, what do? They cause the LDL to be absorbed into the cell, the cholesterol in it to be absorbed into the cell. If the receptor is not working, or if the receptor is not there, then you have a lot of LDL in the blood. صح? So what statins do is they increase the receptors in the wall so that more LDL cholesterol is absorbed so that there's less in the blood. That's how it decreases the blood cholesterol levels, okay? So it increases cholesterol in the cell, but it decreases it in the blood. Is that clear? That's one way it can statins can help. The other way is by inhibiting HMG-CoA reductase so that you can't synthesize mevalonic acid, okay? That's good. Where was I? Okay, so familial hypercholesterolemia. You're going to hear about this disease a lot more in the future too. It's mainly known as an autosomal dominant condition, but there are some cases like this, for example, its mutation is recessive, okay? So this is an example, and it's under the same, it causes the same disease, okay? But most other diseases under familial hypercholesterolemia or other mutations under hypercholesterolemia are autosomal dominant, okay? So what's the difference between having high cholesterol in the blood as compared to high? Isn't it bad either way? Cholesterol in the blood, why it's bad is because it can build up. It can build up in the walls of the vessels, and it can then cause atherosclerosis. It can precipitate in it can cause like many different things, you know. But in the cells, let's say they can be stored or they can be mobilized. They go to the liver where the liver it's excreted, you know. But in the blood is if there's so much cholesterol in the blood, then it can actually end up with the diseases like atherosclerosis. I hope this cleared it up. But in cells, I think of it as it being stored there rather than being in the blood, okay? Is that clear? Okay. Then what was I saying? معلش, my brain freeze شوية. Okay, so familial hypercholesterolemia. It's mostly autosomal dominant. So we know in autosomal dominant يعني إيش? Only if you just have one copy of the gene mutated, that's enough for you to have the disease. Okay, so you have heterozygous FH and homozygous. The FH is the heterozygous form is it's rare, but not as rare as the homozygous one. Okay. So it's 1 in 250 actually. There are other diseases that are much more rare than this, okay? So what it causes is hypercholesterol levels and premature coronary artery disease, and it's usually due to mutations like LDL. There are many mutations, but an example of it is an LDL receptor defect, an LDL apolipoprotein, okay? I don't think you need to know the details of these things. What you need to know is that there's a heterozygous form and a homozygous form. The heterozygous form, it will cause the patient or individual to have premature coronary artery disease and hypercholesterolemia. But the homozygous form, this one, it's more severe because they have both genes mutated, so they're going to have very high LDL cholesterol levels. So it's going to be worse for the patient. You're going to have these patients. They present with heart attacks in their childhood. Okay, that's how severe it is, and that's mainly what you need to know. بصراحة. I don't think you need to know the details. I added some details about the genes that are associated with it. There's like 4 genes in the notes, so you can read about them. But I think you need to mainly know that the homozygous, it's autosomal dominant mainly. The homozygous one is more severe. And this one, yeah, it's LDL receptor. It's the same thing as well, what I just said, because an LDL receptor, if it's deficient or defective, then you can't uptake cholesterol. So you'll have high cholesterol levels in the blood. Now the treatment. So the treatment for the heterozygous form, for the homozygous form, I think there's dietary and all this. It probably won't help because it's so severe. So I think it has it has a different management plan. But for the heterozygous form, dietary interventions, weight loss, exercise, these are important things. These are generic things that you need to keep in mind. They're usually very good for lowering cholesterol levels. Also cholesterol lowering drugs like the one we just talked about, statins. Okay. So in combination with diet, they cause upregulation of LDL receptors. So they increase the level of LDL receptors on the wall, and that helps with decreasing the LDL in the blood. Okay. This is just for you to know, the most powerful statin can cause a 60% reduction in LDL, so it's pretty good. There was something that okay. Once I remember it, I forgot the question. It was a good question. When I remember it, I'll ask you guys. Okay. Cholesterol and bile synthesis. So this is how cholesterol is converted to bile acids. Okay. First of all, as you guys mentioned, bile, it's secreted in the intestine. Okay. And it's a watery mixture of phospholipids and bile salts. And what are bile salts? They're basically conjugated bile acids. Okay, so let's begin from cholesterol. First, cholesterol is converted into a bile acid. Okay, then once the bile acid is conjugated, as in you add a glycine or a taurine amino acid, okay, it becomes called a bile salt. Okay, that's basically what you need to know. And a conjugated one has glycine or taurine as an amino acid. Now, this process happens in the liver. Once the bile is made in the liver, it goes and gets stored in the gallbladder. And then whenever you eat and you need, bile needs to be excreted, it can be secreted from the gallbladder or even directly from the liver. Okay. Let's see, what else did I want to mention? More is in the next slide. Okay, but yeah, what you need to know is that once it gets conjugated to glycine and taurine, it makes the bile acid more soluble. Because if you think of it, the bile is going to be excreted into the intestine where you have lots of fluid, lots of food, and everything needs to be soluble there in order to help emulsify the fat. Because its purpose is also to take care of the lipid soluble products and emulsify them, help with digestion. So it needs to be soluble enough. Okay, that's why it's conjugated with glycine and taurine. Okay. Okay, yeah, that's it, I think, for this one. This picture, I added it so that you can help imagine. Yeah, okay, it's for explaining this. Okay, so I'm going to explain it on this. Okay, so here is the liver. It synthesized cholesterol by de novo synthesis. It gets converted to the primary bile acids, which are these. These are the primary bile acids: cholic acid, cholic acid, okay? The ones that aren't conjugated yet. And the ones that are formed in the liver. So they're called primary bile acids. Then they get conjugated by glycine and taurine. That's when it's called the bile salt. So the primary bile salt then gets excreted or secreted actually into the bile duct. In the bile duct, it gets secreted, then it goes to the duodenum. So then you ate, okay? Bile is secreted, it emulsifies the fat and everything, it gets broken down, gets deconjugated. So glycine and taurine they get freely absorbed, and the primary bile salts become bile acids. Then the bile acids, the primary bile acids, they either get reabsorbed again, or they get converted to secondary bile acids and absorbed. Okay. Some of the bile acids, either the salts or the bile acids or the free cholesterol that's in the bile, they get excreted through feces. Okay. So what you need to know, Fihina, the bile salts become deconjugated in the bile acids into bile acids, and then they're either absorbed or decarboxylated. So this is what makes the primary bile acid into a secondary bile acid when it becomes decarboxylated. Okay. Then the secondary bile acids are absorbed and they're reused. Any primary or secondary bile acids absorbed, they become reused, they become deconjugated, they become conjugated again, and then they get secreted. Okay, and this is the cycle for bile. And as I mentioned, a lot of the bile, or some of the bile, like 0.5 grams per day, they get excreted, and that's how you get rid of the cholesterol. Okay. Any questions about this slide? Let me see. Yeah. Basically, what is it? Okay. So now bile salt deficiency. Okay, cholelithiasis. This is something you're going to hear about even in fourth year. It's a very important disease, something that you're going to have to know about. The first thing you need to know is that cholelithiasis basically means gallstones. Okay, whenever you see the word chole, you think of the gallbladder. Lithiasis, stones. So if I say if you see the word nephrolithiasis, that means kidney stones. Cholelithiasis, gallbladder stones. Okay, so The reason we have gallstones, or why gallstones exist, is when there's so much cholesterol, okay, in the bile. So it's due to increased biliary cholesterol excretion. And this happens when you have high cholesterol, for example. And there are other causes too, like obstruction in the biliary tract and other things. You don't need to know about them right now. But yeah, increased biliary cholesterol excretion, this is one of the main causes of gallstones, and that's what you need to know. And then the gallstone, what's more tricky or whatever, the gallstone, the smaller it is, the worse it is, or not really the smaller it is. I mean, if it's small enough to leave the gallbladder, so this is a gallbladder filled with stones, okay? If it's small enough that it can leave the bladder, it can then obstruct the bile duct, the common bile duct, and that's the worst condition. Then it can cause like it can cause inflammation, it can cause jaundice, it can cause your liver function tests will be high, you'll have a liver problem, you can have infection, then it can have fever. So as soon as it causes this obstruction in the bile duct, that's when you need to do cholecystectomy. That's when usually the patient comes to the hospital, and they usually have right upper quadrant pain, and it comes usually when they eat. They might even have a fever. If it's inflamed and there's infection, he would have a fever. He can even have jaundice if it's kind of obstructing the common bile duct. He'll have jaundice because of all the bile. Okay, you don't need to know these details. I'm just saying them because for your own information. But just know that once it causes obstruction, that's when it usually needs a cholecystectomy, where they remove your gallbladder and the obstruction. And yeah, remember gemfibrozil or gemfibrozil. Remember it? It can cause it. Okay, it can cause increased biliary cholesterol excretion, because he could bring it in a clinical scenario. Patient who's taking. He came presented with pain that comes after he eats. He's been taking gemfibrozil for some time. So what is the cause? Or he can say, what is it? I don't think he'll ask you for the treatment. He'll just more ask for pathophysiology or what could be the cause. Okay, so you need to link them all together. Okay, now steroid hormones. This is the fun part for me. So steroid hormones are very, very crucial hormones in your body. You have many, they're important for carbohydrate regulation, like glucose, for example. They're important for mineral balance. So the level of sodium and potassium in your blood, it can be affected by mineral corticoids. And reproductive functions, like secondary sexual characteristics, these are also affected by steroids, okay? And beside each one, you can see the steroid responsible. So gonadal steroids are like what? Androgens, testosterone, estrogen. They are related to reproductive functions. Mineral corticoids, they're related to like aldosterone, for example. They allow mineral balance. And glucocorticoids, like cortisol. Those are the main ones, And when do you have steroids in the blood? So usually steroids are increased in the blood whenever you're going through a stressful situation, whenever you have pain or any disease or chronic illness, that's when your body starts producing steroids, okay? And you need to know that they're also, since they're made from cholesterol, they're not water soluble, okay? And as I said, if anything is not water soluble, it needs to be either bound to something or packaged, okay? So in case of steroids, they're always complex to specific binding globulins, okay? So for cortisol, for example, it has the corticosteroid binding globulin. For testosterone estradiol, it has the sex steroid binding globulin, okay? That's, if you guys have any question about these slides, if I'm going too fast, just let me know, okay? Okay, so glucocorticoids, mineral corticoids, androgen, these are all the examples. I don't remember if I wrote this or the doctor did, but these, you need to remember these names, okay? Cortisol, aldosterone, we're gonna be talking about them. Testosterone, estrogens, you have estradiol and estrone, and progesterones, like progesterone, okay? This just needs memorizing. Okay, so how are steroid hormones synthesized? So steroid hormones, as I said, they're derived from cholesterol, and they only differ, like the steroids, the different steroids, they differ in the ring structure and the side chain that's attached to it, okay? And they're not, because they're so lipid soluble, they're not packaged or stored inside of the cells. As soon as they're synthesized, they're immediately released, okay? So because they immediately diffuse across the membranes and leave, literally, as soon as they're synthesized. That's why your body does not synthesize the steroids 24/7. It synthesizes them when they're required, when the cell is stimulated to synthesize them, that's when it makes it, and that's when they're released into the blood. And where are these exactly synthesized? In the mitochondria and smooth endoplasmic reticulum. Okay, if you remember cholesterol, do you remember cholesterol where it is synthesized? In what organelle? No, okay, liver, but no, where? It can also be synthesized in other tissues like adrenal cortical. Okay, I'll repeat the slide. But no, I want to know if you guys know where the process of de novo synthesis of cholesterol happens. Okay, someone answered it in direct message, but I want to see if anyone else knows. Where does de novo synthesis occur? Cytoplasm, right? Okay, so it happens in the cytoplasm. So in whatever cell, it happens in the cytoplasm of the liver, but it can also happen in the cytoplasm of any steroidogenic tissue, any tissue that makes steroids or cholesterol has de novo synthesis of cholesterol in the cytoplasm, okay? But it mostly happens in the liver, okay? But it can happen in the adrenal gland, in the testes, ovaries. They also produce cholesterol. They're not going to wait to get cholesterol from the liver. They'll make their own cholesterol as well. They'll make it where? Also in the cytoplasm. But then the cholesterol in the cytoplasm, once it's made, it goes to the mitochondria and smooth endoplasmic reticulum, where it forms steroids. Okay, so that's what I wanted to get at. That steroid hormones, they're formed from cholesterol, where? In the mitochondria and smooth endoplasmic reticulum. Okay, and as soon as they're made, the steroids, they leave. Okay, as soon as the steroids are made, they leave the cell. Okay, they don't stay stored in the cell. That's what I was saying also for the person who asked me to read the slide. That's what I was saying. You know, once the steroid is formed, it doesn't stay, it doesn't get stored in a vesicle. It leaves directly the cell because it's lipid soluble. It just leaves the cell. Okay, and what I wanted to say as well is that because as soon as it's formed, it leaves. It doesn't. Your cells don't produce steroids 24/7 until they're stimulated to do so. Okay, so like our adrenal glands, our gonads, each gland in our body, it has a hormone that stimulates it to produce something. Like how we said insulin. It stimulates cholesterol synthesis in the liver, صح? So when it's stimulated to form, then when the cell is stimulated to form, that's when it makes cholesterol, and that's when it makes the steroids, and that's when the steroids are released. Okay. So like when you're in cortisol, when you're in a stressful state, when you just had, for example, people who are going through any chronic illness, like I'm trying to remember this, like IBS. I don't know if you guys know IBS or IBD, or any GI illness, even psychological stress, even pain, any illness in your body, it increases cholesterol, because cholesterol helps you deal with stressful situations and with stressful disease conditions, basically. Okay. And yeah, I'm trying to see if there's anything else here. These are the steroidogenic tissues that you need to know. These are the only tissues in your body that produce steroids. Okay. Adrenal glands. You guys know where the adrenal glands are? Where can they be found? This is like an anatomy question. Inside the kidney? Where? Yep, on top, above it. From its name, you can kind of guess, like adrenal, I guess it means above. I don't know, probably in some Latin way or something. But yeah, adrenal glands, they're on top of the kidney, and they're responsible for producing mineral corticoids, glucocorticoids, and androgens, okay? And gonads, like the testes and ovaries, they produce the testosterone and estrogen. And placenta in case of pregnancy, it also produces, if I'm not mistaken, progesterone, okay? And I think it produces estrogen as well. But yeah, that's during pregnancy, you don't need to know it. What you need to focus on is the adrenal glands, okay? Yeah, okay, next slide. Like, this is a very simplistic view of the process. By the way, I rearranged some of the slides here just so for the flow of information, okay? Okay, so here I put the functions of each below each one, because that's important for you to know the function, so that when we discuss the disease, you can understand it better, okay? So one thing you need to know is that progestin, it's the precursor for all these hormones, okay? Cholesterol first gets converted to progestin, and then progestin can be converted to either glucocorticoids, mineral corticoids, and as such. Glucocorticoids, the zibda of all this is that glucocorticoids increases your blood levels of glucose, amino acids, and fatty acids. Okay, remember these things. Glucose. So how I think of glucocorticoids is that it's also the opposite. It's kind of the opposite of insulin. It works with glucagon. Both glucagon and cortisol, they increase blood glucose. So that's how I remember it. يعني إنه glucocorticoids, they increase blood glucose. They increase protein synthesis in the liver, and because you need protein synthesis in the liver, for protein synthesis you need amino acids. So it increases amino acids in the blood and transportation of the amino acids to the liver. Where does it get the amino acids? It breaks them down from other places, from proteins and other tissue. They break down the amino acids, they go into the blood, go to the liver, promote protein synthesis in the liver. And it also mobilizes fat in any other place. It mobilizes fat so that there's free fatty acids in the plasma, and then they use the free fatty acid for energy. That's what glucocorticoids cause. Mineral corticoids. Okay, so for mineral corticoids, they cause renal reabsorption of sodium and secretion of potassium and hydrogen ions. Remember how we said in the mineral corticoids, they are responsible for mineral balance, maintaining the balance of minerals. And from its name, you can kind of guess. So it helps reabsorb sodium. So what can you guess happens after you cause sodium reabsorption? I'm trying to hide what's written. So what happens when a mineral corticoid increases sodium reabsorption? Yes, water retention, leading to what? What happens to your blood? When you have water retention? More hypervolemia, because technically mineral corticoids are only released when you have dehydration. So basically it increases your blood volume and increases your blood pressure, yes. If you have excess mineral corticoids, then you will have hypervolemia and hypertension, okay? But yeah, it basically increases your blood volume and your blood pressure. And it causes, if it's in excess, it can cause hypokalemia, okay? Which is it's a serious condition. I think it can cause arrhythmias, if I'm not mistaken, okay? Then androgens. Androgens, from its name, it's a hormone that is responsible for male sexual characteristics, okay? So high levels of androgens in a female can cause masculinization, okay? Can cause her to have male pattern hair growth. She'll start having more hair around here, around the beard area, mustache, etc., okay? So it promotes masculine features, basically. So in a male, it's responsible for secondary sexual characteristics, and in a female, it causes growth of the pubic and axillary hair, okay? And in the male too, obviously. Androgens can then be converted to estrogen, and that's what's usually used in the females for secondary sexual characteristics, okay? So these are all the steroids, and I think we discussed, oh yeah, one more thing. Glucocorticoids and so steroids, since they all come from one origin, yani cholesterol and progestin, their functions kind of overlap. So glucocorticoids, the example being cortisol, it's mainly glucocorticoid, so it mainly has glucocorticoid activity. But at some point, it can have mineral corticoid activity, okay? It also has some mineral corticoid activity, but not as much as its glucocorticoid activity, okay? Same thing with the mineral corticoids. They're mainly mineral corticoids, but they have some sort of glucocorticoid activity, okay? They kind of overlap, and that's mainly because they have the same precursors. So they're very structurally similar, so they can kind of bind with each other's receptors, okay? But remember, in the end, that the glucocorticoids, their main function is they're more stronger with their glucocorticoid activity than their mineral corticoid activity, and vice versa. And this is a bunch of explanation about how the androgens are made. They're made in the adrenal glands and the gonads. In the males, they have dihydroepiandrosterone, DHEA. It's the main one formed by the adrenal gland. So for glucocorticoids, we have cortisol, the main one. For mineral corticoids, we have aldosterone. For androgen, the main one is, I call it DHEA. It's basically dihydroepiandrosterone, okay? And the had by the adrenal gland. The testes, they secrete testosterone, dihydrotestosterone, and androstenedione, okay? You can read that on your own. Okay, now steroid hormone synthesis. This is all about, okay, so this is about the same thing, how it's formed in the mitochondria and endoplasmic reticulum. You mean this, the things that's written in a note? You don't need to know, you should read them for your understanding, just to help you understand, okay? But what you need to know is mainly what's on the side. Hatta this, by the way, I don't know if he'll ask you about all the functions, just understand it, yani, and know what it causes. I think he'll care more about this one for mineralocorticoids because it's linked to the diseases, yani, and I'll repeat them anyway with the disease. And this you should know too as well, and this here. But notes, it's mainly for your understanding and to explain the slide better, yani. Okay, so steroid hormone synthesis. So as I mentioned, in any pathway, you have a rate limiting step. And the steroid hormone synthesis pathway, the rate limiting step is not an enzymatic step. It's the step where the cholesterol, okay, is transported from the cytoplasm to the mitochondria, okay? This step is the rate limiting step. It's the slowest step in the pathway, okay? And who's responsible for this step? It's the steroidogenic acute regulatory protein, okay? So this is considered the rate limiting step because, like I mentioned, cholesterol is formed in the cytoplasm, but the steroids, in order to form the steroids, it needs to go to the mitochondria and endoplasmic reticulum. So its transfer from the cytoplasm to the mitochondria is the slowest step. It's the rate limiting step, okay? So remember, that's the rate limiting step. And remember that it needs who's responsible for the step, the steroidogenic acute regulatory protein, okay? Yeah, this explains how cholesterol is synthesized, how it's made from acetate. It can come in the steroidogenic tissues. How do they form cholesterol? They use acetate, but they can also use cholesterol esters that are stored in the cell already. They can break it down, basically remove the fatty acid, and there you go, you have cholesterol. Or they can take cholesterol directly from LPLs, lipoproteins. Lipoproteins taken up from the plasma are the most important when steroidogenic cells are chronically stimulated. Just remember that when steroidogenics are stimulated for a very long time, their main source of cholesterol becomes the lipoproteins. And yeah, this is basically what I explained. Okay, so this is a very detailed process of how steroids are, it's a pathway for how steroids are made. You can see here we have cholesterol. Okay, let me see. No, I don't like this one. Okay, I'm just, okay, there. So here we start with cholesterol, okay? Then when does cholesterol start to go down this pathway? When it's stimulated by ACTH. Here I'm talking about what? This is a process that happens where? In the adrenal gland, okay? Okay, Dr. Raja, not to worry about this. Okay, khas, perfect. That means you don't need to understand you don't need, sorry, to memorize the steps. I think that's what she means, صح? You don't need to memorize the steps. Okay, good. What I wanted us to explain is that it's stimulated, this whole process is stimulated by ACTH, because this, I'm pretty sure she might want you to know it. It's related to what we're going to talk about later, okay? So the adrenal gland, it's stimulated by ACTH in order to produce steroids, okay? Like I mentioned before, it won't produce steroids unless it's stimulated, and it needs ACTH, okay? So ACTH is a hormone that comes from your pituitary gland. Pituitary gland, it's in your brain, it's actually like right behind your eyes. It's in between here, okay? That's just a fun fact for you. But you know, ACTH, once it's secreted, it goes to your adrenal gland and stimulates this process. You need to know about this enzyme. Actually, it's supposed to be in other slide. No, this is 17. This is an enzyme. Yeah, this is 17-hydroxyprogesterone, because you need to know about the enzyme defect here and here. Okay, we'll talk about them. This slide, I just wanted to show you ACTH, okay? This is a more simplified slide. You can see how cholesterol becomes pregnenolone, then progesterone, and as we said, progesterone, it forms the basis for the others, okay? Okay, so if you see how there's like multiple enzymes. Here, عندك 21 alpha hydroxylase, then you have 11 beta hydroxylase, okay? This is for making aldosterone, so it needs these two enzymes. For cortisol, you need the same thing, 21 alpha hydroxylase and 11 beta hydroxylase. And then for androgens, you mainly need 17 alpha hydroxylase. So if any of these enzymes were missing, let's say 21 hydroxylase was missing, then the progesterone can't be converted down, it can't go this pathway, and it can't go this pathway either. It will have to go to the androgen pathway. So it will produce androgens. And then your body will be like, okay, there's no cortisol or aldosterone. I need to stimulate the adrenal gland. So you'll have a lot of ACTH secretion. It will stimulate the adrenal gland, but then again, the adrenal gland can't form cortisol or aldosterone. So again, it will form androgens. And the cycle keeps going. So this person ends up having what? So much androgens and little to no cortisol or aldosterone. Okay. And this is something that happens in a disease called congenital adrenal hyperplasia. Okay. This disease has it has multiple causes, like in genes, gene-wise. So it can, a defect in this enzyme can cause it, or a defect in this enzyme can cause it. Okay. And yeah, we'll actually discuss what it means. But right now, what I want you to know is that when one enzyme is missing, the pathway gets directed to the whatever enzyme is there and available. Okay. So yeah, she wants you to know these two enzymes, 21 hydroxylase and 11 beta hydroxylase. This is the other name for it. I think she wants you to know that too. And as we said here, 21 hydroxylase, you needed to make deoxycorticosterone to make corticosterone and aldosterone, and you need to make 17 alpha hydroxyprogesterone to make the 11 deoxycortisol and then cortisol. Okay. Now we'll talk about, yeah. Okay, so as I mentioned, congenital adrenal hyperplasia. In case, okay, let's first talk about if the 21 alpha hydroxylase was affected. Okay, so of the 21 alpha hydroxylase, yeah, they're very important. Yeah, because honestly, even in fourth year, yeah, in our pediatrics rotation, they still ask us about this. So it's definitely important for you guys to understand. It will save you a lot of time in the future, definitely. Okay. So whenever you have 21 alpha hydroxylase deficiency, first of all, you need to know that there's two forms of this deficiency. It's either completely absent or it's partially absent or partially defective. Okay. And as you can guess, yani, the complete absence, it's more severe than the partial one. So if there's complete absence, when do you think they would present? When do you think the patient would come to you? If they had complete absence of this, do you think they would come to you earlier or later? At what age? At birth. Yeah, so at birth, or maybe, yeah, they can even come at birth if it started quickly, or even within a few days after birth, come on. And they mainly come with what? Salt and water loss and hypovolemia and shock. So let's take a step back and try to understand why this happens, okay? Here. Okay, so this is the same and the slide that we had earlier, but I put an X on the pathways where you have 21 alpha hydroxylase. So this patient, he had complete absence of 21 alpha hydroxylase. So he has no cortisol and no aldosterone. If you have no aldosterone, what's going to happen? Think of the function of aldosterone. What does aldosterone do? You'll have dehydration. This was a sign. Okay, so yeah, so you don't have mineral corticoids. You're not going to have aldosterone. You're not going to be able to reabsorb sodium, so no water retention. So you're going to have what? Low blood volume. And what? Low blood volume leads to what? Hypotension. So they can even have hypovolemic shock. Okay, that's why they said they can present to you with shock. Other than that, it's called salt wasting. Why? Because he's going to lose all his sodium in the urine. Okay, that's why they call this form the classic form. It's called classic classic congenital adrenal hyperplasia is the salt wasting form, because they have no 21 alpha hydroxylase at all. So they have no way of like reabsorbing the salt and the water. So they just lose all the salt in the urine, and they end up with hypovolemia and shock. Okay, and this is called the adrenal crisis. Okay. And that's what I said here. They present with severe dehydration and salt wasting and all of that. Another thing, they'll present with something else. What other thing will they present with? Can you guys guess? Inflammation. I get it, because cortisol is anti-inflammatory, but no, that's not really the case. It's something else. What's going to happen in here in this pathway? If you don't have 21 alpha hydroxylase, what's going to happen? If it's a baby girl, for example, or let's say if it's a baby boy, what's going to happen to the baby boy? How will he present? He's going to have a lot of what? He doesn't have 21 alpha hydroxylase, so he won't have corticosterone, he won't have cortisol. What's going to happen to this pathway? Androgens, yes, he'll have a lot of androgens. So if it's a boy, he'll present with very early signs of puberty. Okay, so it's called precocious puberty, where they sense that where the person gets puberty at a very early age. Okay, because of all the androgen. And in a girl, you'll actually see when you examine the baby girl, you'll see that the external genitalia looks more like male external genitalia. It's not completely male, but it just has some masculinization to it. Okay, because there's actually another, and this is interesting, by the way, if you search up androgen insensitivity syndrome, this is a disease where actually a girl with XX chromosomes, okay, she comes out with completely male external, or sorry, it's the opposite, where a boy, he has XY chromosome, he comes out with female genitalia, external genitalia. When you look at his genitalia, you say, okay, that's a girl. And he grows up living as if he's a girl. And then they find out later on that he's actually a guy. He was all this time, he's been a guy. And it's called androgen insensitivity syndrome. So you should look that up. It's actually interesting. A lot of people, even in Saudi, they find out that at a later stage that they're actually a guy and they've been raised up as a girl. They look like a girl. And yeah, it's interesting, صراحة. But yeah, what was I saying? Androgens, because they have high androgen level, the female babies will have some masculizing features in her external genitalia. Okay, let me go back to the slide. Okay, yeah. So the boys will have early virilization, early hair growth on the baby, and the girl will have masculinization. This is called, okay, the one that I just explained here, the salt wasting one, the one that's freeze with shock, they present in the early few days of life. This is complete deficiency. Okay, this is called the classic form of congenital adrenal hyperplasia. The partial enzyme defect, this is the late onset form. This is the non-classic form. This is the one where the person, he grows up normally, there's nothing wrong, he just comes later with what? Usually it's not a he, usually it's the girl comes. Usually the girls come because they have menstrual irregularity and hirsutism. Then when they do, why does she have menstrual irregularity and hirsutism? Because she has so much androgens. Okay, if a guy has so much androgens, you won't really notice because he grew up, it's part of his sexual development, they won't really notice. They'll just say, oh, he got early puberty, but no, it's not that early to the extent where he's a baby, because it's only partial enzyme defect. And he won't have the signs of hypovolemia or shock or all that, because it's not that severe. But in a girl, no, she'll have hirsutism because of the androgens. So obviously she'll come to you and complaining of that, and then you realize that she has the partial enzyme deficiency of 21-alpha hydroxylase. Is this clear? Is this slide clear? Because this is important. You need to know the difference between the two forms, the non-classic form and the classic form. All clear, guys? Yes, clear. Okay. So, okay. Now the congenital. Okay, so this is the same thing. This is diagnosis. Okay. So can you give us the main differences? Okay, so main differences. Okay, so imagine if you're in the classic form and non-classic form. Classic form, you have complete deficiency of 21-alpha hydroxylase. Non-classic form, you have partial deficiency or a defect in the 21-alpha hydroxylase. The idea is it's not, you don't have as much deficiency as you have here. In the classic form, you have complete absence. In the non-classic form, just slight absence, okay? So a deficiency, I would call it deficiency. That's the first thing, difference in the availability of 21-alpha hydroxylase. The second thing is the way, when do they present? The classic form, they present as early as at birth or a couple days after birth, okay? While the non-classic form, no, she can present in her in her teens, for example. She presents later. Okay, the idea is, no, it's later. Okay, and now the third thing is how they present. In the classic form, they present with hypotension, dehydration, they're salt wasting, they're losing a lot of salt in the urine. The female babies, they have masculinization of the external genitalia. The males, they'll have early virilization. While the non-classic form, usually it's a female that presents with female that presents with menstrual irregularities and hirsutism, which is male pattern hair growth. Okay. I think that's it. Yeah, that's pretty much it. Because yeah, so that's the main difference between the two. And another thing, and it's mentioned here, by the way, as well. In هنا partial enzyme defect. It's a late onset form, only menstrual irregularity and hirsutism in young females. Okay, so diagnosis. This is important. How do they diagnose it? They look at the levels of this. Where is it? Here. I want to go to the picture. Yes, this one. Okay, that's why I highlighted this, because you're going to have high level of this since you don't have, this pathway is blocked. This pathway is blocked. So you're going to have a lot of 17 alpha hydroxyprogesterone, and because of this blockage, so they measure this 17 alpha hydroxyprogesterone in their blood, and when they find it and it's very high, that's how they know, okay, this is she has congenital adrenal hyperplasia due to 21 alpha hydroxylase. Okay. Yeah, that's it. I think what else? Yeah, they find increased plasma 17 hydroxylase as early as 4 days after birth. This is in the classic type, since he said as early as 4 days after birth, it will be very high. Okay, and it will cause, yeah, precocious puberty in boys. We know this too, since they have lots of androgens. What's the effect of low cortisol? So low cortisol would be, if you think of it, it would be low glucose. What else? He'll have low lipid levels. He won't have high lipid levels. But the thing is, there's other things that compensate, if I'm not mistaken. Like it's not the primary feature of the disease. That's why they don't really mention it that much. What has a bigger effect on it is the aldosterone. You get me? So that's what you really need to remember. But cortisol, they don't really mention it that much, its effect of having low cortisol, because it's not as significant as the aldosterone one. Okay. Cortisol, by the way, what you need to know more about cortisol, for future use, is the diseases where you have lots of cortisol, like Cushing's syndrome, Cushing disease. You'll take this later on. That's when it actually becomes critical. That's when you need to know the functions of cortisol, because it's actually very common here too. I think you'll take it in endo-repro probably. But yeah, I hope I answered your question. Now 11 beta hydroxylase deficiency. Okay, so let's look at this one first. Here I block the pathways with 11 beta hydroxylase. So what's the difference in this compared to the one before? Here we have 11 deoxycorticosterone production and 11 deoxycortisol, because the 21 alpha hydroxylase is present. Okay, but the difference is the presentation. So when 21 alpha hydroxylase was blocked, we didn't have deoxycorticosterone. So we had almost no mineral corticoid activity. But now in this type, where you have block of this, we have 11 deoxycorticosterone available, which does have mineral corticoid activity, but it's weak. It's really weak. Okay, but if you know, okay, so it's weak, صح? So because we still don't have aldosterone formation, what's going to happen? ACTH will induce the gland to produce aldosterone, but it won't end up producing aldosterone. It will end up producing 11 deoxycorticosterone. So eventually we're going to have what? Very high levels of 11 deoxycorticosterone. And at very high levels, it's basically able to cause significant amount of water and sodium retention. Okay, so that's what you need to know. 11 deoxycorticosterone, it's weak when it's it's weak. Okay, but when you have very high levels of 11 deoxycorticosterone, it's able to cause a lot of water and salt retention. So how do you think a patient with 11 beta hydroxylase deficiency would present? If he has he's going to have a lot of 11 deoxycorticosterone, which I said causes water and salt retention. So what is this going to lead to? Edema. Okay. You're thinking more of extracellular fluid space. What about the blood? Because the retention is going to happen to the blood, like in the kidney, you're going to reabsorb the salt and water from the urine into it's all going to go to the blood. Hypervolemia. Okay, so hypervolemia leading to what? His blood pressure, basically. What will his blood pressure be? Hypertension, exactly. Always link those two together, by the way. Usually whenever you have hypervolemia, it ends up leading to hypertension. Hypovolemia, hypotension, okay? Okay. So yeah, that's what it's mainly going to cause. That's the difference between the 21 alpha hydroxylase and the 11 beta hydroxylase. 11 beta hydroxylase, you have some mineral corticoid activity being formed, and that's what leads to that's what leads to the a lot of water and salt retention, hypervolemia, and hypertension. Okay. What's the opposite of shock? You tell me, Faiza, what's the opposite of shock? Shock is basically when you have, it's a systemic, because there's so many types of shock, but one of them is hypovolemic shock, when you have low blood volume and hypotension, okay? So the opposite would be hypervolemia and hypertension. So yeah, I don't think there's a word for it, if that's what you're asking. Okay, no problem. Okay, so 11 beta hydroxylase, this is the slide he wrote. I think I added, by the way, this stuff. Not sure, did I add this or did I do something? I don't know, but yeah, you'll see. So yeah, you'll have 11 deoxycortisol. You'll have high levels of deoxycorticosterone, which, as I said, it has mineral effect. You'll still have mascularization in males and early virilization in females. Again, because you're still going to have androgen production, right? Not this, sorry, here. You're still going to have a lot of, this pathway is going to be stimulated a lot because your body is trying to produce cortisol, okay? So it's going to be stimulated a lot, but you're not going to be able to produce cortisol nor aldosterone. So you're going to have high levels of this, and high levels of androgens, okay? So they will still, in both of them, they will have virilization and masculinization, okay? In both the 11 beta hydroxylase and the 21 alpha hydroxylase, okay? That's why I said it's important to know the functions of androgens, okay? Okay, so despite inefficient production of aldosterone, yeah, as I said, you will have the effect of 11 beta hydroxylase, which will cause 11 beta HADA, the effect of 11 beta OH, which is basically the one that I mentioned, the deoxycorticosterone, which will cause the hypertension. Okay, so it's a weak, yeah, this is basically what I said. You'll have salt retention, volume expansion, and hypertension. That's what you need to remember. The other one will cause salt wasting, hypovolemia, and hypotension. And I think that's it. Yeah, that's it. Do you guys have any questions? I hope it was helpful and you guys actually understood. And if you have any questions, you can email me or send me a text message, Adi. And yeah, no problem, guys. Good luck. Make sure to study MOL weekly. So yeah, could we have the same presentation notes? Yes, I can send it to the development GND, and they can send it to you guys. They have contact with you guys by email, so they can send it to you. And yeah, is it okay if I ask you something about the first program? I don't know if I can. Sure, you can ask. You know what's a better idea? Send to the Spirit email, and we can respond to you there. Okay, about first. If for those who don't know about first, you guys should try to apply. It's a really good program. It will help you in Spirit next year. So good luck. I'll send it to the Mesa. Okay, no problem. Okay, I think that's it.